and then I applied to some places. Because of my earlier exposure to the crystallines, I got a position with this guy called Professor Sci'versation. Again, another very aggressive mentor, very aggressive. I mean, I was fortunate to work with him, but my return to India was a kind of very unplanned and unscheduled because I was thinking of working there for at least 3 to 4 years. Certainly, I was planning to come back, but I came a little earlier than that. Anyway, during this period, I worked on on the correlation of domain structure with the chaperone activity of alpha-dystroglycan, which happened to be one of the small extract proteins, by using the single tryptophan mutants. That was a new approach at the time, but now these days people are not doing this because we have got better technology to do that. So while doing this was my main project, like studying the domain structure, relating it to the chaperone activity with the single tryptophan mutants. I had about half a dozen single tryptophan mutants of alpha-dystroglycan. But while doing that, I also worked on structural transition involved in the enhanced chaperone activity of alpha-dystroglycan because by then Mohan Rao and many other people showed that alpha-dystroglycan with the temperature has a higher chaperone activity. And we provided the reason why it happens. So but that I could publish in JBC. But the first part, I did not publish. You may laugh at me because I had to, come back to India to take this position here. At that time, I submitted this to EMBO journal, and they have asked for some revision, which so Ravish will not compromise without doing those experiments. I could not do that experiment. Then that paper is still there in my computer and probably in his computer. So that was my return to India. Well, it was Surolia's lab and Suravi's lab and the group that I had, it was a notorious group in Surolia's lab. So not only science, but we did all sorts of other things, that gave me an opportunity to have good friends and learn many things from my life. Whereas in Cleveland, it was a very tough job for me to survive. Though I could do excellent in the lab, but it was a very cold place. At times you will have 20 feet of snow because of lake effect. I was living about 1 kilometer away from the space, but still coming in this snow every day, walking about half a kilometer, another half a kilometer, of course, inside the building was not easy. I don't know how I survived. I can't imagine now, but I survived and I came back. But that period has actually gave me a lot of learning for my life as well as for myself. I came back, probably I'll tell you why I came back. I came back to India in the middle of 2000 and joined at the same institute where I am now and where I did my PhD. And this is my journey in a nutshell. Like, at the time it was named as a senior research officer, but it is nothing but scientist C. Well, this is a journey from scientist C, D, E, F. Now currently I am G. But when I joined as a scientist C, I was given a lab like this, a small place, which is much smaller than the office right now I am sitting here. But we managed. Then slowly, I know it was one of the things for me to is build my lab. So you could literally see that here we were building the lab. We built it, as I said, which is much smaller than the office that I'm sitting in my office now. So I had some visiting appointment during my 20 years here. Like in 2003, I went for 5 months to University of Michigan. Then again in 2005 and 6 for another 6 months. Like this, there are 4 or 5 different visits to these places, which actually gave me an opportunity to learn and to come back and to implement certain things that I learned at these places. That was the best thing that I could do, and that happened to me. Well, these are the research areas that I am in now with my group of the people. One of the things is, as Prash said, I was working on crystallines. When I came back to India, I did not know anything but for working with a few proteins, whether it's lectins or crystallines. So I had to start some work immediately. So I started that. One of the things that later we expanded on the same thing is protein quality control systems and small extract proteins. And slowly I initiated some work on bioactive molecules, which I am going to elaborate a little bit for the benefit of the people. And I also work with the different animal models. That became one of the major part of my research. And this is the main thing that now I am into it because this is a nutrition institute. I realize that unless otherwise I work on the core of nutrition, my purpose of working in this institute is not going to be justified. So that's how this thing is started. So if you want me to summarize how I do, the area is molecular nutrition with respect to some age-related disorders. I'll show you what are those age-related disorders. We try to understand some aspects of molecular basis of the disease, and we try to understand the nutritional aspects of it, and we also try to evaluate the potential of functional foods with respect to these diseases. And while doing so, we get some opportunity to understand some of them as a biomarkers of these diseases. I use both the in vitro system, all the in vitro systems, animal models extensively, and of course, we also work with the human studies. So it is like a complementing with all the things, which really helps us to make few points in a much better way than using one single system. So since Prash asked me to share my work in 5 to 10 minutes, I don't want to do all the things because it's not a scientific talk. With respect to protein quality control system, small eats of proteins, what we did basically we provided the information on structure, function, and the expression of small eats of proteins. There are about 10 small eats of proteins, including a couple of crystallines under hyperglycemic conditions, and then how these aspects, whether it's structure, function, or expression, can be modulated by dietary and nutritional factors. Why I decided to work on hyperglycemia or diabetes is that around the time I think the heat started about the diabetes in India. So I also decided that okay, that is one area that one should get into it. So I really feel that I took a right decision. I'll come to it a little later. Then we also showed the differential expression of small eats of proteins in various target tissues of diabetic complications and provided new evidence to understand the role of these things and so on and so forth. While doing so, we could show that one of the small eats of protein, that is HSP27, could serve as a potential biomarker of diabetic nephropathy as a part of one of the PhD students. Uh, this happened, and but now this only gave a lead, but we want to validate this and see whether it really serves as a novel biomarkers or any better things that we want to identify. And our recent studies suggest that altered ubiquitin proteasome system and ER stress, and of course decreased autophagy, muscle atrophy, retinal degeneration, and neuronal cell death under obese conditions and diabetic conditions. So again, we have shown how diet restriction and other modulation by other dietary means. So this is what we have done, and through this work we could publish 40 to 50 papers and about 5 6 PhDs. Till now, every time one student works on some of these aspects. So there's a line of work continuously that happens in my lab. This is another thing that, as I told you, I decided to work on diabetic complications because when you are talking about diabetes, actually the complications are most important than the primary aspect of the diabetes. Well, I don't have to tell you the numbers because currently there are about 500 million people in the world, and which could go up to 600 by 2030. India has anywhere 80 million people. So this uncontrolled or prolonged diabetes leads to various complications, literally involving every part of the body like eyes, kidney, heart, brain, etc., etc. So these organs, when they get affected, they are the main reason for the morbidity and mortality in diabetic patients. So in addition, the metabolic syndrome or obesity also more or less leads to same set of complications. For that matter, overlap of these complications. So that's why we decided to work with a set of complications of diabetes, such as retinopathy, which affects the retina, cataract that affects the eye lens, nephropathy that affects the kidney, and of course neurodegeneration. Now, the first thing what I decided to work on this because while doing my PhD, I was familiar with a couple of the metabolic pathways. I told you, I worked on the metabolism of the lens. So I fairly familiar of the metabolism of the lens. So I decided to take up a couple of the pathways that I am familiar with it. Because if you see the molecular pathways that are linked to diabetic complications are mentioned here. One is polyol pathway, other one is protein glycation pathway, which is in short, which we call it as advanced glycation end products pathway. Well, there are a few other pathways, but I am familiar with these two things. So I thought, okay, I should try to understand what happens under diabetic complications. How can I use these molecular pathways for targeting developing new molecules? So let me tell you what happens in these pathways. In the polyol pathway, the glucose gets converted to sorbitol by an enzyme called aldose reductase, and this is one of the culprits in the diabetic complication. Similarly, the excess sugar and sugar derivatives that you see in diabetic conditions will modify various proteins, and these highly modified proteins are called advanced glycation end products. So this is also one of the problems. Therefore, in addition to other pathways, these two targets serve as a drug development targets, etc. So inhibitors of aldose reductase and anything that prevents these modifications will also be useful for for preventing or treating the diabetic complications. So in this background, we decided to work with the functional foods and bioactive molecules with a program where we have screened and at times we isolated and characterized the novel molecules. So basically to screen a lot of functional foods and wherever it is possible to identify bioactive molecules. So these are the systems we use for for the first pathway we have cloned all those reductase and we have got a recombinant protein and you can simply do the enzyme assay and screen as many as you want. Whereas for protein glycation system, you can prepare the modified proteins and you can try the molecules that you want to test whether which one is going to prevent this modification. Later, I try them in some kind of ex vivo systems, whether it's organ culture or cell culture. Then finally, we test them in animal models before you really try to see whether they work for the human studies. Okay, I'll go through it very quickly. So you could see that in the last 10 or 15 years, we have created quite a lot of dietary sources. I think close to almost 150 or 160. I'm not putting all of them here. Using that recombinant human alloreductase assay or Ratlan's islands assay. So we have identified some of them which have got the potential. Here we have used varieties of extracts. I'm not going to go it because it's not a scientific term. And identified some of them as as ones that seem to be promising. One of them happened to be gooseberry or amla that we call it as, or in an official name, in scientific name is Emlica officinalis. Then we became curious what is there in this to that is inhibiting this reaction. So we are using that bioassay guided fractionation. We could identify a novel molecule. I mean, this molecule is known in the nature, but in amla, we have first time we have identified a molecule called glucogallin or gallite glucose. By all this methodology, I will not take you through this. By HPLC fractionation, subsequently by mass spectrometry, then characterized by both H1 NMR, C13 NMR, even elemental analysis, because that is the first time we are showing. So you want to make sure that that is the molecule you have identified. Finally, this is the glucogallin that we could find in this source, that which has got the potential to inhibit this aldose reductase. So this happened to be a little bit more specific compared to other related aldoketo reductases. That was one of the problems in the earlier studies people who worked with aldose reductase inhibitors. Because though they have developed aldose reductase inhibitors, but some of them are not very specific to the enzyme that we were interested. It's AKR1B1 or ALR2. Luckily, this one happened to be more specific to, or relatively more specific to this one. Then we have shown what kind of inhibition. One thing that we could do is that this is all simple enzyme assays. We have to really prove that whether it works in a system. So using a AR or aldose reductase transgenic mouse, which can be grown, the lenses can be cultured in high glucose, where you because of the transgenic expression of aldose reductase, you see a lot of sorbitol. And the same thing if you do with the molecule that you have isolated, that is beta glucogal, you see whether this suppression can happen. Yes, the one that we have identified is suppressing even in a high glucose condition. That says that it is inhibiting aldose reductase and therefore it is suppressing the sorbitol accumulation. That is fine. But this is something interesting, though I am not an expert in bioinformatics or modeling and docking, etc. But one of the students did this. The previous studies on all those reductase inhibitors were like this. They were small molecules. They were sitting on a anionic pocket. Actually, the active site is like this. It has got two pockets. One is anionic pocket, other one is specific pocket. But the earlier ones were, a very small. It was an open kind of. I mean, once you have only small molecule here, it gets closed. This is called closed inhibition. Whenever you have a better molecule which occupies both the pockets, it is called open inhibition because it does not allow the second pocket to get closed. Luckily, the beta glucagon happened to be like this. It was not allowing the closing of the second pocket, so it is an open inhibition. And then we also decided, okay, if you were to add some more moieties like this, I think it may even impart better specificity. That's what we actually designed some novel ones. The synthesis is going on. We tested it, but some work is still going on. The point here why I have taken so much of time is that this has given a new direction to aldose reductase inhibitors. That one is it has to be specific to this one. It has to be a bigger molecule than what they earlier were, so as to occupy this both the pockets, so that you may get better inhibitors. Well, like this, we have also worked with some other sources. I will not take your time. From this, we have also synthesized analogs, and some of them we have even patented. The other part I was telling is, preventing the modifications, which is not that simple like what I was showing with the enzyme essay, where you need to modify the proteins. You can take any model protein, as I listed here. You can take any protein for of your choice, collagen or lysozyme, modify with any of the sugars. Then you see whether these modified proteins can be evaluated in the methodology, because you need to use at least 4 or 5 different complementary methodology, because it is a very non-specific kind of essay if you take only one essay. Therefore, one has to show it in 4 or 5 different essays, such as non-critical and fluorescence. Upon modification, you will get crosslinks. You can also identify crosslinks. You can even quantify the modified protein. Then you can detect these modified antigens by immunoblotting, etc. By using all that methodology, we could screen some of the functional foods and dietary sources, identified some of them, which I have listed here. So I picked up 1 or 2 of them and to characterize what are the molecules that are there in this. We came up with one molecule in cinnamon that is called procyanidin B2 by all the methodology, which I will not bore you. So this happened to be a molecule that is present in cinnamon, which is preventing this modification. Likewise, we have also identified a molecule called ellagic acid and rutin in different other sources. Well, finally, we want to validate them in a real system or a physiological situation using an animal model. Here we create or induce diabetes in animals. Then we allow these diabetic animals to live for some time so that they develop some of the complications that I mentioned, either cataract or they develop retinal degeneration or they develop renal problems. And then we test these molecules that either the isolated molecule or the source that you have isolated from it. So you can see when isolated, is it going to give the same activity or is it better or less? So anyway, all those things we have done using these animal models. We have got the tools to evaluate all these complications. We could show that, as I said, we have identified some of these things like, procyanidin B2, beta glucogallin, and then everyone knows curcumin is there in turmeric. We also done some work with it and then protein. So we could show with these things the delay of diabetic cataract, amelioration of diabetic retinopathy. I'll not show you all the things what we have done here, and amelioration of diabetic nephropathy. So at the end of it, I could summarize this as like we have screened the functional foods and characterized in some of them with the novel molecules such as beta glucogallin or procyanidin with aldosterone inhibition or anti-glycatic activity. So then later we validated this in animal models to indicate the potential of these molecules. Now, one of the things that we decided to do is that because all these we were working with each of them individually, because now we know that some of them work synergistically, and also if you target 2 pathways, the the potential may be much better. Therefore, we are combining both aldosterone inhibitors and anti-glycating agents, and then making a formulation which we call it as a polyspice formulation. Right now, we are working with this polyspice formulation based on our earlier studies of almost like what we have close to 10 different publications with these molecules. Now, we're using this knowledge and how to mix them, what proportions, how long, how can we assess all that. We are now at the end of it, we want to come up with a formulation which can be used for the prevention or arresting of the progression of diabetic complications for human purpose. Well, I used to work in the lab earlier days, but off late I am not spending that much of time to work on the bench. But once in a while, I still spend, just to show that I am working. Okay, we have good number of women force in the lab. At times, we do celebrate Women's Day because we respect them a lot, and we also encourage them because they have got unlike we boys, they have literally many other commitments. It's not easy for them to survive in this such career. So one of the things I make it a point to encourage them, help them as much as you can, so that they also come up with their potential and then their interests can be fulfilled, their aspirations can be fulfilled. Other part, as we work with these diabetic models, I told we also work with obesity model, etc., etc. So one of the things that happened in our institute is we have spontaneously developed obese rat model. I don't know. Luckily, instead of doing all that, experimentally creating obesity, etc., it just naturally happened. And luckily, this obese model also has got a problem of cataract and retinal degeneration. So we have characterized that for the first time because of the obesity, they have cataract, they have retinal degeneration. Now, we use these models to show that they have got neurodegeneration, and then we want to test our functional foods or for that matter any nutritional things. Most importantly, we even did the dietary decision on these animals. We could show some change with this with respect to these complications. The last part is the most important thing for me. That is that in the journey, this is where now I entered into this another area through a different means. Actually, this is something interesting I want to share with the young people who are listening to me. That sometimes you need to pick up the leads and make your road. One of the things I was telling, the orbital pathway that people work in animal models, in cell cultures, in vitro studies, etc., etc. When we had a couple of all those reductase inhibitors or functional foods, to work with it, I decided to see whether this can be translated to human purpose. When I discussed with some clinicians, they said, actually, what is the status of this pathway in humans? Has anybody published in India that what happens to the CLR activity and polyol pathway in diabetic people with complications? Without complications? No, we did not have it. So then I decided to do this study. You could see that, we took normal people, we took diabetic people without complications, we took diabetic retinopathy people here. As expected, all those detected activity is more in diabetic retinopathy, and the product sorbitol is also more. But this is an average of 200 patients' data. And one of the things, I am not shown that slide here is that, all those reductase or for that matter, sorbitol has to be accumulated at certain level. Then only it creates the problem. So for that, the activity has to be at some level, like, at least above 9 units or above 6 units. So therefore, I decided to see out of the 200, how many have the activity above 9. Out of 200, only 20 happen to be above 9 units of the activity. Actually, this gave another direction for us to proceed further, that all the failures of all earlier aldose reductase inhibitors could be because of this reason. Could be. No, I mean, I could be wrong also, but I suspect this is one of the reasons. The reason is that if you give all those reductase inhibitors to this group where the enzyme activity is less than 3 or less than 6, it's not going to make any difference. Maybe that's the reason it did not work in these patients. You need to choose the patients who have got literally high aldose reductase so that you can actually see the effect of it. So, but we are yet to do it, and we have to prove that point. It's only at this point of time it is hypothetical. But that actually gave a different direction to me, or a curiosity that why these 20 people have got more than 9 units of the activity. Well, with my little bit of biochemistry, I could relate it to one of the pathways in the intermediate pathway, that is pentose pathway, where transcriptase is the one which competes for the NADPH with the same enzyme that I am working, aldose reductase. So for some reason, transcriptase is like inhibited. The most of the NADPH can be utilized by this enzyme. One of the reasons is if you have B1 deficiency, transcriptase activity goes down. Then I suspected, is it possible that these patients would have less of B1? Luckily, it appears to be that way. That the lower the B1, higher the aldose reductase. That means when there is a less of transcriptase, more of aldose reductase because of the B1 deficiency. And this actually helped me to get into a different area. So there is a possibility that your nutrition, particularly micronutrients, can influence all these metabolic pathways that are linked to the diabetic complication. Therefore, we actually decided to a study like this. So we literally we have done with a study with this many patients, normal people, diabetic people with without any complication. Then we also have people with retinopathy, neuropathy, nephropathy, and then we try to understand what happens to the nutritional status, such as status of vitamins. In diabetic retinopathy, we could find the deficiency of B12, whereas in diabetic neuropathy, we could see the B6 deficiency and B2 deficiency in addition to B1 that I showed earlier. But there was no problem with the fat soluble vitamins. Then again, I come back to the same thing that I try to see why the deficiency of these particular micronutrients can affect the complications. Already I showed if you have B1 deficiency, that can actually shift the NADPH to aldose reductase. Whereas if you have the B12 and B6 deficiency along with folate, it can shift the metabolism in such a way that homocysteine get accumulated. And also B6 deficiency can actually add up to the accumulation of advanced glycation end products that is known earlier. So now I wanted to actually prove that point. You could literally see here in this study, when you have the deficiency of B12 in diabetic retinopathy people, you see the inversely increased amount of homocysteine. A nice correlation. Lower the B12, higher the homocysteine. It is known, but in diabetic retinopathy, in our context, we are showing that. Similarly, this is very interesting, more than that, because it is somewhat known somewhere else, but we have proved the same point here in our context. But this is more important. When you have less of B6, there's a possibility that more of the modified proteins are accumulated in the system. That is what we showed, but we have to do a lot of work here. But that has actually given a chance for us to do more work in this area. So just summarize this, I told you B12 levels were low in diabetic retinopathy, B6 was low in neuropathy. Then, because of the B1 deficiency, there is a possibility of ALR2 sorbitol accumulation in retinopathy. And if there's a less of B6, there's a possibility of adverse glycation end products accumulation. This is what actually we have come up. Now, this association will not help us to make a big claim unless you prove it. Therefore, we decide to work with the animal models where we create, this particular deficiency, whether it's B12 deficiency or B6 deficiency, with or without diabetes, and see what happens to all these complications. Right now, we are working on this. We did some. We have published with respect to B12 deficiency, but we are yet to do a lot more work in this area. Well, sometime when you are at this stage, we have some publications. People do recognize you, and then I was so happy to deliver this Dinesh Chakravarti oration because he is a man who actually initiated and encouraged the people in India to do eye research. So on his name, Indian Eye Research Group has a oration that is Dinesh Chakravarti oration, and no less than a person, my hero, Balu, could introduce me to this oration, and then I was very happy to do this. Actually, the earlier people you see the oration Chakravarti orators, I feel I'm very privileged to be in the list of the Chakravarti oration. But that again, led me to a different direction. That when I was talking about B12 deficiency, I had to decide. Okay, you are talking about B12 deficiency in diabetic retinopathy people, but what happens in the normal population? So we decided to understand the micronutrient deficiency in apparently normal people. This is a very recent study where we show that it's not only B12 deficiency, you have B2 deficiency and B6 deficiency. And you remember, these are supposedly healthy adults. This is done in and around Hyderabad on 300 people. They're supposed to be healthy and apparently normal, but you have as good as 50% prevalence of B2 deficiency, B6 deficiency. So that tells that you need to screen the people to really know the subclinical deficiency. Otherwise, most of them will not result in a clinical condition. And then we know that one of the reasons could be lower intake of these nutrients, but one has to really show that is it because of the lower intake. For that, we have to develop some tools. We developed a tool called raw food based quantitative food frequency questionnaire. So I write this in one sentence, but it took one PhD to do this. We developed this tool. A student called Sarah, she has developed this tool. This is a part of her PhD. But now later, many people use like another nutrition student, Shalini, few other people. Using this tool, we would come up with what is the adequacy of these micronutrients in our population. Of course, this is a small sample size, but we are doing it in the bigger sample size now. So we show that how many people take the required amount, how many people have inadequacy with respect to these nutrients. So while doing this, some people also look at, and this has got a lot of relevance for the nutrition research and society. There were a lot of news items about these studies, and then seeing one of these studies, the then Lokaayukta Justice Subhash Reddy got interested about our studies. And one day, his secretary called me when I was in a seminar. I'm calling from Lokaayukta's office. Justice Subhash Reddy wanted to talk to you. All of a sudden, I was so much terrified why I got a call from Lokaayukta. What did I do? I said, Madam, I'm in a seminar. I'll talk to you later. When I called him back, he said, I read your news items about your micronutrient deficiencies. I am very much interested to know that what is going on and all that. One day, he came even to my lab and he gave his sample for all his nutrients. We did it. So, why I'm saying, sometimes it happens like this. And then when he came to our institute, of course, he passed away last year, but because he was Lokaayukta, he came with a big security and they came to my lab. Everybody got doubt what Bhanu did, and there's a security in his lab, and Lokaayukta is here. Did he do something wrong? Later, this is what has happened. Well, more or less, I'm done with I didn't like summarizing my work, but I need to say a couple of things here. There are few people who, apart from Surolia, Suravich, and of course, Surolia is I can never forget him for what all that I learned from him and what all that he did it to me. But then these are the people. Professor Mark Petras, right now he's at University of Colorado, Denver. Uh, uh, while I was working with Suravich, we were collaborating with Mark Petrash on crystallines. But when I came back to India, I I asked him because when I was expressing my recombinant aldose reductase, there's a stability problem. I was not getting enough amount of that protein and all that. I asked him, "Can you help me?" Then he helped all the things. He sent the clones, he sent the bacterial clones, he sent the stabs, everything. Well, we managed, but after some time we said, Banu, don't waste your time. I'm going to ship you the recombinant all those days. My technician will verify in as much as you want. You just go ahead with your studies. That's how actually we could do all that studies. He was so generous, and then I still continue to work with him on many aspects. The other person, the first time I went to a visiting fellowship to Radha's lab when she was at the University of Michigan, that is the time I expanded my research to retina. Before to that, I did not even know how many layers retina has got. Believe it or not. But the reason she gave me a 4 or 5 months visiting fellowship was that at that time she identified a novel mutation in one of the proteins linked to retinal degeneration. She wanted to do me some protein work because she knew that I know a little bit of protein. You believe it or not, the 4 or 5 months I did all the things I could do, but it was an utter failure. I could not help her anymore. So I spent 5 months in. But that actually 5 months of period helped me to understand a lot of things about retina, and then along with her and in collaboration with her, we even had a Indo-US program with her, and I initiated retinal degenerations. Now we know a lot more things. We could see how you test the retinal functions, what is retinal degeneration, or what types of all those things. So if I were to have an elder sister, I don't think so she would have been much better than Radha. So that is what I regard Radha. Then another person is my first graduate student, Satish. He's a different guy actually. I don't want to take your time, but then sometime in 2014, I went to his lab on a DHR fellowship. At that time, because by then he had a lab and he was working on pulmonary fibrosis. But at that time, my interest was to see fibrosis in other issues like the retina or particularly kidney. Because I am working with the diabetic kidney, I wanted to see whether can I do something in that area. I went to his lab. I worked in his lab 2 months, and then so many things have happened. I will not take your time, but a couple of things I can tell you. It is collaboration. What all that we did, particularly the molecular regulators and therapeutic approaches for fibrotic diseases. One of the things that he did is that Wills tumor one, which is a transcriptional factor, he uncovered this a pathogenic role for this WT1 in fibrosis, which promotes the fibroblast activation and then peripheral areas of the lung. A part of it is recently published in EMBO Molecular Medicine. Of course, a couple of other papers in JCA Insight and Journal of Immunology. That more than that, of course, these are all his own projects. So we were part of it. Like a couple of my graduate students worked there. But because of that, now I am embarking on a project which is very close to my heart: role of aging and hyperglycemia in multi-organ fibrosis, particularly renal, and therapeutic scope based on a glycating mediated thing. Because my area is protein modifications, how this modified proteins can initiate the fibrosis. If so, can I use my agents that I developed in the lab, anti-glycating agents, to ameliorate or modulate these pathways? Right now we are working on, I think some interesting stuff is happening. One of my graduate students is in his lab at present, but Satish is one guy, he will not compromise. I've been asking, like, hey, we have some data, we'll publish. He says, no, 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 you need to generate some more data. I am not going to do that now. So we have to wait for some more. Well, apart from this, I also have to acknowledge one person who just retired a couple of years ago. He has been there with me ever since I joined in NIN as my technical colleague. He was a backbone in my lab. Of course, now we have other technicians. We will, I think, excel him or rather, do much better than him. But he was a backbone of my lab. Literally, almost every PhD student learned something or the other from him. But later, they tried their own things. But initially, they had to spend some time with him, and he was a man of perfection. And many times, even I learned many things with him. So that's why I could, focus more on my projects and more on my publications, etc., etc. Well, there are many other people. I know more than maybe 50 people if you include all the project staff, other things. It is difficult for me to name. in the 20 years of journey. Everyone did their bit, and each one of them are good in their own way. You know that when you have 20 people, one is good, one is relatively better. I don't want to say that. Therefore, everyone did their bit, and we had nice time. Uh, Probably this is the current group. Now I'm showing it. Of course, even Lopa retired last year. Now I have got a couple of new technical colleagues who will, I think, take up the same job that what my earlier technical colleagues did. But more than anything else, these guys have contributed to this entire journey and the publication that whatever we thought. Even in my PhD degree, at that time there was no issue of ethical thing. There was nothing like ethical committee, animal ethical committee. I could go to animal house anytime and kill as many as rats I wanted. But these days it's not possible. Still, teachers for our life, they sacrifice their lives. Well, you can also get a chance to have snaps like this with vice president or sometimes with minister of science and technology. That happens in your journey. And then this could not have been possible, as I'm telling, in a single group or a single individual cannot produce this kind of work unless you work with various groups and people like Radha, Mark, Satish. Then I also have a national collaboration with Nagini, Dhirendra Katti from IIT Kanpur, Madhulika and Indrapal Singh from IIT Chemistry Department and Biotechnology Department, ISCT. Then my clinical colleagues, they were just, they never bothered what I'm doing, what whether I'm publishing or not. Whenever I said I want to do this, okay, if I have a chance to give the sample, we will do. That's what they did. Now also I have working a project with Prashant, and my recent collaboration is with another graduate student, that is Anil. He's another an extraordinary guy, very enthusiastic guy. Well, I also work with these institutes because I'm not putting the names. I work with more than one people in these institutes. Therefore, I have collaboration. And at the end of it, the, measurable outcomes. If I have to say, well, these are the publications with this metrics. And then currently there are 6 graduate students, 4 PDFs, and then these are the students who have graduated from my lab. I have put this slide because I want to say that more than anything else, I'm very much happy and satisfied with this because most of them are now like kind of into academics or research. I've already talked about Satish. Now he's due for professorship. Now he actually, is my mentor. It is no longer my mentee. He is my mentor. Even Anil, hopefully will become my mentor. Now, Srinivas, PNB is an excellent guy. Of course, he is in Aurobindo Pharma. He could have been in a real academic setup like this, but for various reasons, he shifted to there. And then, Muthu is assistant professor in State University. Kishore is in the state. Sudhakar is currently a scientist and in the same group. Then I think Sneha has still to make. She took a career break because of a kid, but I think I'm trying to see how can I make her to get into the career. And these guys are at different places doing excellently well. I'm so happy. I'm not taking any credit, but I just want to say that I am associated with these guys, and I learned a lot from them. I don't know how much I trained them, but I learned a lot from them. In addition to that, a few postdocs out of 6 or 7, 3 of them are well settled. Now Srinivas is again in the same department as a scientist. He came to my lab as a postdoc, worked for, believe it or not, 10 years. Then at the end of the 10 years, he got a position. Then Raghu is another excellent postdoc. Now he is a research scientist at Texas University, so and so forth. Well, in addition to that, we also do a few other things. I contribute to the recently released dietary recommended dietary allowances for the Indian people. I did my part, and also recently we published an interesting paper. This is again a kind of dream come true publication for me. It took 20 years for me to publish a paper in American Journal of Clinical Nutrition. Again, it's not a primary data. Those secondary data, I'm very much happy because it is going to provide some information that the vitamin A supplementation, universal supplementation that we are doing for the last 50, 60 years. This paper tells us that there is a review is required there. I think you need not to do universal supplementation. Maybe we have to focus only in the areas where vitamin A deficiency is really there. So that is the one. I think it is going to create a lot of debate. It is in press. Once it comes out of it, we have tough time to answer the question, but it is going to steer the vitamin A supplementation program. I'm saying it again. The last part I'm again happy is that I have a passion to go to the schools, colleges, and to the community and talk to the people, tell about the science, and encourage the kids. By different routes. One of the things that I do is science education or nutrition education or awareness program to school kids and college kids, even common people. As a regional coordinator of Telangana Academy of Sciences, as a member of NASI local chapter. Earlier, I was a national mentor for Inspire program for school science projects. Now, I also work with a couple of NGOs. One of them happened to be Vande Matra Foundation, whose focus is, empowering and improving the standards of government schools in rural areas, particularly the education sector. But for this pandemic, on average, at least one day in a month, I used to spend, or sometime more than that, maybe 2 days. But the pandemic stopped. So this is what happens. You see, Sneha was showing the DNA isolation to the kids. They are so excited to see their own DNA. And at times you could. This is my own school where I studied. These kids demonstrated some experiments. You could see it is in another school. And this foundation, because of the excellent work, not because of me, there are 4 or 5 dedicated people who work in this foundation. And we were given an award for the excellent job by the Science and Technology Minister in Delhi a couple of years ago. My own kids, when they were young, they were also doing some experiment. They were so curious, they were doing some projects at home. And Basant was doing something in the lab also, and Anurag was doing some circuits. But now they are grown up. I don't think so. They are any more interested with this thing because they have got only 2 gadgets. One is smartphone, one other one is laptop. So, but for that, I don't think so. They are interested anymore with this kind of science. Okay. Well, finally, it is possible for me to talk to you, show all these things because of the two wonderful people who give birth to me. And I don't know, my father, since childhood, I am becoming emotional. He had tremendous confidence and faith in me. I don't know what made him to think. He never stopped. Whatever I wanted to do, he helped me. Even when he did not have money, he said he will arrange some way or the other. He allowed me to do what I wanted to do. I think that's the reason I'm here today. And this is my extended family. We are 2 brothers and 1 sister and kids. Finally, these 3 people allow me to work in the lab much without much complaints. They do complain, but not much. So that's how I am here and I'm talking to you. Thank you, guys.
Thank you so much, sir. It's an incredible journey. I can completely sense the kind of emotional drive that you're going through. I was also a bit emotional, for many reasons. It's been a terrific presentation, sir. It's an
Thank you.
awe-inspiring presentation.
Thank you.
Friends, many of you don't know that Sir Banu sir is differently abled, and despite all the odds, those things has never touched his course. He would always perform his experiments. He's always willing to do whatever best that he could do. And the final setting is absolutely great. The final slide, when someone gives due credit to his parents, that speaks high volumes about that particular person. The most important thing about Banu sir is he gives no credits to his students, which is absolutely a kind of a rare breed. Seldom we find people acknowledging their students or considering their students as mentors. Have you ever come across any such people or any such persona? At least not me. I think many of you would definitely agree. This is Sci'versation. It's absolutely very very inspiring. Thank you
Actually, I must
so much,
thank
sir.
you
I have
for
several questions. We'll
I
also
must
ask
thank you
some
for
questions.
providing a platform like this to share my thoughts because I always talk science in a different platforms, but this is slightly a different thing. It gave me an opportunity to share some aspect, not all of it, of course. Thank you, Prash.
And not all, sir. Not all, yeah. So we are right on it, sir. So one of the important things that I wanted to ask you is you have worked extensively on alpha crystallines, and you have wonderfully explained how you came across that particular thought and other things. But considering the fact that there is a kind of chaperone-like function associated with alpha crystallines, they could there could be viable biomarkers that could be associated, and probably because of that chaperone-like function, probably cataract can be I mean, we couldn't expect cataract in some kind of substitution. But in India, if you really look at that, there are a lot of micronutrients that are not viably associated with any person. We are either deficient in vitamin D or vitamin A, or vitamin B, vitamin C, or whatever. So how important is epigenetics attributed to such things, sir? What is your take as a nutritional genomicist on this?
So you are asking for with respect to the crystallines or otherwise also?
Yeah. I mean, of course, for alpha crystallines, for the first thing, because you have been working for over 2 decades. Yeah, please.
Yeah, the alpha crystalline or small exact protein stories like this. I was not working for my doctoral work. Around that time, alpha crystalline belongs to the small exact proteins is known. But around the time, one guy called Harvich in the University of California, he showed that alpha crystalline can also function as a chaperone by simple aggregation essays and all that. And many people picked up. And at that time, Mohan Rao did a lot of work in India. He was known for that. Then at the end of my PhD, because I was working with the crystallines, I also did some work, and then I kept that passion with me because I knew how to do it. But then by then I completed my PhD. I went to Suroli's lab, but Suroli asked me to do something on lectins. But then I also told him I have some passion with it. He allowed me to isolate alpha-crystalline and all that. But he had something, he assigned some major work. I was focusing on that. Luckily, when I was applying for a postdoc, this guy Vitol Suravich took me. So again, I could do that work there. And when I came back here, I started this work, particularly on the modification. The direction that I took is that alpha crystalline S works as a chaperone. But what can happen when a person is diabetic, or if it is you have high glucose, the chaperone itself can get affected. That was the concept actually. So I was not working on the actual chaperone function, but my focus was: is it possible that this function can get compromised? So that's what we did. A lot of work actually. At least 5 or 6 papers are there. At the end of it, we wrote a couple of reviews saying that this is one of the areas that you can target. That means you prevent the modification of alpha crystalline chaperone. It will take care of the other crystallines. That's what actually we have published. But translating it into the actual clinical situation is not that easy. We're trying to see in animal models we could show that. But how do you do this in humans? We are still figuring it out. One is like there are mutations to this alpha crystalline, which can affect the chaperone function. But in addition to that, you also have these modifications. Therefore, if you I'm talking about the anti-glycatic agents in generally, not necessarily for alpha crystalline. It can also target alpha crystalline so that it can protect alpha crystalline, and in turn that takes care of the lens transference. That is one part. But the micronutrient deficiencies and epigenetics is the real scenario in India. I think there are many people who use the word. I don't want to use. That's why I use the word called molecular nutrition. The nutrigenomics. Everyone writes papers and reviews, but in India still nutrigenomics is in infancy. But this is the place where nutrigenomics has to be done in extensively. I think we just started it, and hopefully at different places. Unfortunately, perhaps one thing in India, nutrition research is not fashionable. Maybe recently some people are, but people think that nutrition. Oh, come on! Where do I get a paper in Cell and JPC and all that if I work in nutrition area? That is one thing. And also the big institute, the so-called big institutes, also for some reason or the other, they don't focus on this. There are some groups. I don't want to belittle them, but by and large, the so-called the premier institutes in India are not taking or putting their thoughts into the nutrition research or micronutrient deficiencies. The basic research, when I say, with respect to the epidemiology, you have plenty of information, but you have to get to the bottom of it. Whether it's epigenetics, how they influence the genome, how they interact with the genetic variations is a big area.
Yeah, well said, sir. I mean, this is absolutely very good. I know, I mean, when it is, whenever, vitamins or nutritional genomics is considered, I mean, people also have some kind of stepmotherly treatment towards that. That's very unfortunate. Yeah. So moving further, sir, I know, getting publications and performing experiments, validating, the kind of research that you have been doing, would take ages. But it's incredible that, you have garnered to produce more than, sometimes, 15 to 20 papers a year, and that's not a kind of an easy thing. So how difficult was it for you, or how easy for you, sir? Because this, I mean, a strong message should go to the young generation, especially the current PhD fellows, who really need to take inspiration from you on how you could afford all these kind of things despite all the odds.
Well, both. One is it is easy because the people who work with me, who came to me with some hopes and some aspirations, and I told them that no, this is the only way that we can show it to the world that no, we are there. So they worked. So they generated the data, and it is difficult. The second part because I am not convention educated, neither my language command is not so good. That you have thoughts, but it won't flow on the paper. So it takes some time. So I still remember, me and Satish sitting next to each other, framing each and every sentence. But over a period, we grown. It became a routine stuff, like on average. Now, actually, whether somebody likes it or not, in a lighter sense, I tell everyone in the lab, per head per year there should be one publication, whether you like it or not. Somebody say what do you do with the publications, it's fine. Now somebody say what do you do with this many, it's fine. But for these young guys, that gives a hell of a confidence, hell of a confidence. Once they have a paper of their own, that okay, I did it and I published it, I know it. Because during my 4 years or 5 years of my PhD, I could not publish my papers for various reasons. When I went to Surolia's lab, I had 24 hours at my disposal. I used to sit after 11 o'clock and I wrote four my PhD papers in his lab, sitting in the night, because I realized how important having a publication. Otherwise, nobody looks at you. Secondary thing is what difference it makes is now let's say having 50 or 100 publications, one or two may not make a difference, but in the beginning, it makes a difference. It gives a lot of confidence.
Wonderful, sir.
Well, it's
Well
difficult. It
said.
is easy and it is difficult. Even now, you could have seen in 1 hour how I talk in English. It's not easy for me to write, but I work hard.
Right. True, sir. Well said. Well said. Sir, if you really look into the wonderful small metabolites that you have generated from all those diabetic mice and all those experiments, through GCMS and LCMS or whatever, I mean, looking into those particular compounds or identifying genetic signatures that could be responsible for, say, a nutrient intake or micronutrient intake in humans is certainly a need, actually. I mean, especially looking into the Indian perspective. But somehow, we in India, we have not generated those specific genetic signatures so that, we can take this, towards the precision scale. So where are we lagging, sir? Why is that, we are not able to do that?
Yeah, look, there are many factors to it. One is the hi-fi technology that now we call it genomics, whether next generation sequencing or exome sequencing or whatever it is. Those people who have got the expertise, they should actually work with the nutritionist, or vice versa. I actually written a couple of concept notes, and then I don't know if I shared one with you also, that the micronutrient deficiencies, how can
Yeah.
we generate the signatures. And I even shared one with Thangaraj, but now he has become director, it is busy. Sometime I want to sit with him and make a big program on this. But unless these two groups or three groups come together and make a big program, because those expertise and the facilities are not available with us. We do have a platform, but you also need expertise and you need a bioinformatics also. All these things. I think the coming together is very, very important.
No, I completely agree, sir. That's a well said point. I know during your 30 years of illustrious career, 25 to 30 years of a distress career, you might have had a lot
Well,
of
it is not
disagreements.
illustrious like anybody's career. No illustrious.
No, no, we certainly take inspiration from that. So what I was asking is, this is a kind of a common question I usually ask, so that the young generation can take a leaf out of these things. There might be disagreements that you might have made with your colleagues or with your senior colleagues or whatever. So how did you put those disagreements in an agreeable way, sir, at any point of time? Can you give some one example?
Well, quite a few, but now I'm totally a different person. Initially, I used to be very aggressive and very short-tempered. Not anymore, of course, because the age tells you many things. That's where I also had problems, but that helped me a lot. In the long run, people know you, know what you stand for. But at times it creates hell of a problem. Here, once I had hell broke with Surolia. I'll take that example. One day, actually, a couple of days before to the experiment, he gave me an experiment because he believed me. Out of 20 people, he said, we have to do this from morning to evening one day because this protein is very unstable. I want you to generate the denaturating profiles on CD. From morning 8 to evening, we were running like a mission. Then every 1 hour he was coming and checking. Every 1 hour he was coming and checking. Then it was around 6 o'clock. Father, is it over? And he wanted me to do some 4 or 5 different temperatures. I was running, I was transferring from one temperature to other. Then many things have happened. And then all of a sudden I lost my temper. Sir, I can't do it anymore like this. If you come every 1 hour and stand behind me and ask, I can't even transfer from vial to cuvette. No, I just throw out my tray there and went to the hostel. Went to hostel. Then everybody in the lab thought that okay, tomorrow I'm fired because Surya is known for it. If you don't oblige him, you don't have actually your career. It happens. And they thought that okay, tomorrow I'm not there in the lab. Believe it or not, at 9 o'clock, he calls me in the hostel common phone because you don't have the phones in the room. There are no mobiles. He calls me and tells me, Bhanu, sorry, I think I was very anxious to know because that was a new protein and I was very curious to know. I think I annoyed you. And I said, sir, no, 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 it was my mistake because I did not realize your enthusiasm and curiosity, but I was working like mad. So that is one thing. But I also had some similar things with my PhD mentor. I think I don't want to bore you guys. Once I was doing one experiment on UV radiation. This was actually 24-hour irradiation with a 6 hours interval. So if you start at 12 o'clock, next to 12 o'clock, it gets over. So every 6 hours, let's say 6 hours in the evening, 6 o'clock in the evening, 12 o'clock in the midnight, again 6 o'clock early in the morning. So sometime if I start at 11, 5 o'clock early in the morning, you have to take a reading. So I used to come from hostel and do this. One time, the sample that my lab is somewhere, the instrument fluorimeter is somewhere, the exposure is in another lab. So I completed 11 o'clock reading and put my notebook in the lab. And then for 5 o'clock in the early morning, I came from the hostel and straight away I went to the instrument room, took the reading. But I was lazy to go to the lab and pick up my observation notebook. I took a tissue paper and wrote the readings on the tissue paper, left it there, and again locked it and went to the hostel and came back. For 12 o'clock or 11 o'clock reading, again I took my observation notebook, I wrote it. So I wrote it 6 hours, 12 hours, and 24 hours. The 18 hours, it was in a piece of paper, I wrote it later. Then when I saw it, he said, how can you take 18 hours reading after 24 hours? That was his question. I said, this is what has happened. He did not believe. I don't believe you, he said. Then it was like, I gone mad. If you don't believe, I don't think I can work with you. Yeah, it's spontaneously it came. Then, after he's my mentor, why would, his ego is hurt. Okay, if you don't want work, you get lost, he said. I went to hostel, but all my colleagues, the man is here, and and other people came to me, hey, don't do this, man. Don't you want to get your PhD? Come back to the lab. So I came back and did it. But then after coming to my independent career, also I had a lot of problems with the people because I had a different version because these ICMR institutes are very orthodox. Whether you like it or not, I am not blaming anyone. You still have that colonial kind of culture and hierarchy and all that. But at time I used to go to director's office and then there you have to wait and all that. One day or otherwise, you have to come back and go 10 times or 20 times. The people in the director's office are more powerful than anybody. I don't mind. Everyone knows my openness. They appreciate it. One day I told the then director Kamalakrishna Swami, Madam, I sometimes I feel it very difficult. Why don't you put a notebook and I ask for your time, and then at the time you can call me because I don't have to disturb you and you don't have to. Then I simply called her staff. Whenever this boys comes, you don't stop them. Simply send him to my room. But people appreciate it, but not everyone is so generous like her. I had a lot of problems, and I don't want to discuss some of them.
Wonderful, sir. I mean, amazing. It's incredible. So moving further, I mean, we have another couple of questions, but before which, I mean, you have gratefully acknowledged everyone in your career, in your research career, and we have your senior technical officer Yadagiri Garu here, Padur Yadagiri Garu here. So I just wanted to hear from him. What does he think? How does he cherish, the wonderful relationship with you? So, Yadagiri Garu, if you can kindly unmute your this thing and please speak a few words.
Looks like that he lost his connection.
Yeah, I think there was a connection problem. Yeah, and it's okay. So,
I can answer because I think we had more or less same kind of attitude. He was committed for his work, so I think that's where we gelled together. And he also had same kind of problems what I had.
Yeah,
You are there, okay. But before he unmutes and answers, let me tell you, acknowledging everyone is not a great thing. If I don't do that, it's a crime because you might think that I have done it. No, I think everyone has to do that. You cannot live in isolation. You can't work on your own unless people are with you. You are no one.
yeah. Well said, sir. So, Yadagiri Garu, would you like to say a few words how you cherish the wonderful relationship with Banu, sir? Would you please unmute your microphone? Please unmute your microphone, sir. Sir, you have to unmute your microphone.
It is muted.
Uh, you have to unmute your microphone, sir. There is a mic option. You have to click that again. Yeah, please.
Think he seems to have some problem. Okay.
Yeah.
I think I see Sue also is here, but when I met first time Sue, I was so what do you call it? I don't know what to use. With at least I walk with one stick, but he has to walk with two sticks. He goes around the world. So I think that is one message I want to say that it is not your physical ability or disability. Your mental strength is important.
Yeah, I mean, we always look upon you as an inspiration. So that's quite amazing. Even Shiv is also quite amazing. Ever since I've known him from my 2009 days, we are really fortunate, really. So moving further, if you really look into the nutrition as a kind of entity, the current educational policy, the new educational policy, do you think that the nutrition has got a bigger take in that? Because we have almost a bigger change in the way the new educational policy has been carved out. So, what is your take on this?
Actually, I have not gone through the entire components of the nutrition education policy, but I attended a couple of the meetings here and there. Now they are trying to be more practical oriented, including the nutrition part as far as science is concerned. Unlike the earlier, you hardly did some practicals in a plus two, or even in the current situation, plus two is just muggifying and passing the exam. But the new education policy puts a lot of emphasis on learning through experimentation. Hopefully, it will take the shape in the way that it is meant. But I don't know. One has to wait and see. But more than that, there's something called science policy also. Every 10 years or 20 years now, they are coming up with the science and technology policy 2020. I happen to be involved in this discussions of, in two, three forums. And I gave to a group of people, we have given some suggestions. One of the things we suggested is that this new science education policy, science and education innovation policy, has to be taught even at the college and school level. They should know what is the importance of science and technology. And unfortunately, believe it or not, even when I joined at this institute, I did not know that there is a policy like science and technology policy. Only in 2013, when I went for a workshop in Bangalore at National Institute of Advanced Studies, I realized that there were some policies like this. So why I am saying, even the scientists do not know that there is a policy like this, and what do you expect from the kids how to grow up?
Yeah.
So I said mandatorily when a scientist joins, he or she should go through the complete tenets of the science and technology policy, and then the institute leadership should tell them what is expected from you, and they should come up with their vision. Then both have to converge and they have to travel together. Otherwise, I just join and pick up something. I did crystalline work with Suravij, and then I start my crystalline work. Luckily, I could change my direction and work at NIN, but otherwise, sometimes it may not make any sense to the place where you are working.
Yeah, yeah. Very well said, sir. So maybe final few questions, sir. I know you might have answered this in a different way, but the point is more towards nutrigenomics. I mean, if you really look into the ingredients, the nutritional ingredients, they are certainly known to modulate gene expressions, and these dysregulation will probably be associated with different pathways in the human body. So, for example, if at all, if we are to apply such kind of ingredients to our diet, for a healthy lifestyle, for preventing a disease, what kind of changes are specifically needed, and what kind of innovation are we really lacking? I mean, we have wonderful mobile platforms these days. People use smartphones. We use wonderful these things, and yet we feel something is really lacking. So, do you have any say on this?
Actually, I'm not going to say something very, very, what we call different and all that, but we know fairly the nutritional requirements for the people of different ages, like adult or children or elderly people. Well, in India, the evidence-based recommendations are not there, but the hypothetically generated requirements are there. At least they serve a reference kind of thing. The next question is, are we meeting that requirements? So then, to meet the requirements, first you should know that what are the requirements. Even the people in this kind of professional institutions or organizations, how many of us know that what is our requirement? Not necessarily the carbohydrates and proteins, but other micronutrients. Then where from they come? Do we practice that balanced diet or diversified diet? One of the things we recommend from the institute is diversified diet. That means you take as many food items or dietary sources as possible, you will meet all the requirements. The reason is that no single source will give you all the requirements. Let's say vitamin A may be rich in one source, vitamin C may be rich in another source, and then B vitamins are there somewhere else. Unless you take all these different diversified foods, you will not meet your actual requirements. And also with respect to proteins, though now the situation is like this. We have more or less secured the food security. We are reasonably good. Nutritional security is important. I think nutritional security means just having the volume is not what we want. Do we have all the necessary items in that? So that's where people have to exercise their their knowledge. Or if they do not have, they have to gain that, and then institutes have to go out and educate the people. That is one of the things I keep doing. I told at least in a day. I mean, one day in a month or whenever it is possible, we try to educate the people that these are your requirements, how you can meet, and then how you need to focus. Otherwise, there are some fancy ideas. If you take Horlicks, you are the best boy in this world. I don't think so. You have many things around your house and in your locality which are much better than Horlicks and Boost. So, well, I am not against Boost and Horlicks, but this is what we need to do it.
Yeah. So, final few questions, one or two questions. You have extensively worked on aging, and there's also oxidative stress associated with that. And if you really look into the development of biomarkers, there has been a good scope, towards identification of such particular biomarkers, which you have already identified as well. But if you really look 10 years down the line, I mean, as a systems biologist, a systems genomicist, or a bioinformaticist, I think that we could probably take some kind of precision dialogue here. Say, if one subset of population has got these traits, and if these traits harbor these nutritional measurements, can we, in the near future, have a kind of a training population like this and come up with a kind of a prediction? Yes, this is how you need to be prepared for. So, how, I mean, I think your group and probably NIN or ICMR is really doing these efforts. Do you have any say
No,
on
I
this?
think with respect to that aspect, the system biology, or if you want to translate it to nutrition, systems nutrition, I think we are yet to get into that area. But what you said, the most of the things that we give the recommendations or the thing is keeping the population in view. But when it comes to individual, there's going to be a lot of difference from one individual to another. Sometimes these recommendations do not translate it to individual to individual. Now, if you take, apply this systems biology, like say in the US, by this common polymorphism in MTHFR, methyl tetrahydrofolate reductase, C67 to I think I think, where they have this polymorphism, the folic acid has to be supplemented to them. I think about in particular group, I think about 30% of them I have to take up this. Like this, we need to actually come up with some kind of information. We have to develop some panels. Let's say now we are talking about B12 deficiency. Is it because of the intake is less, or even if they take, their absorption is a problem, or their metabolism is a problem with respect to genetic makeup? If you have that panel, you know that whether you need to increase your intakes or you need to do something else. But we are way to go in that direction. We are way to go in that direction. This is where actually systems biology, computational biology, and then a lot of the new tools, that genomic tools, have to be applied to the population and individual levels.
Yeah, perfect. So well said. And so finally, how has COVID affected your lab, sir? Did it affect your research? And if it has really affected, how did you overcome this? And what would be your best advice, to the young generation, in the COVID times or post-COVID times?
I don't know whether I can do the advice or not, but as the ICMR institutes, we were asked to work all through this COVID period. But initial months like April, May, maybe up to June, July, we were asked to work with the 50% of the staff. But we at a higher level, all of us are supposed to come or available all through. So during those 4 months, we worked with the 50% of the force. Like in my lab, that 20 people are there. On any given day, we were 10 people. But from September 1st onwards, they changed that, and we are supposed to work full. But during that 4 months, what I did was that paper I showed, American Journal of Clinical Nutrition. Almost every single day, me and my colleague Raghu were sitting in the lab and then generating analyzing the data, and then writing the paper with a couple of experts, Anurag Krupad and Professor Sachdev. With their help, actually, we did that, and we even submitted a policy document to the government for taking a rethink on vitamin A supplementation. That is the best way that we utilized our pandemic time. We were even coming on Saturday and Sunday and sitting in the lab, because Dr. Sachdev and Anurag Krupad from St. John's, the moment you send, the next day you will get the response. It's up to you to catch up with them.
Yeah,
Well,
that's
advice-wise, it is not. It is individual. One has to have the inside the trust, and then the burning thing for something. No one can force anything on anybody. It is an individual's choice what he or she wants to do, how he wants to do. Okay, if you want to do the thing that can be done in 1 week, you want to do it in 1 day, or you want to make it 2 weeks. It's your choice. Only the mentor can say what happened, what happened. Sometimes if they are irritant people, come on, I'm not going to do anything. You will not be able to do anything. So it is the individual. At the most, you can push it, but not more than.
yeah, yeah. I understand, sir. Yeah. No, that's quite amazing. Sir, I think we should really thank you for your wonderful time and energy on this. You have spent more than 1.5 hours giving an illustrious presentation. It's been a kind of an awesome interview or a Sci'versation. I wouldn't call it as an interview, but a Sci'versation. You have given both of these kind of things. Do you have any final words, sir, before we conclude?
Yeah.
Well, I think in the, the 20, 30 people who have attended this, good number of them, I know them. So and even otherwise also whom I do not know, I must thank them for, patiently listening to my sensible or non-sensible stuff that I've been talking for the last 1 hour. Okay. So, but this is a more of a casual talk, to know that how things work, what worked for me may not work for others, and what works for you may not work for others. One has to figure it out. But then that's what. Uh, Interact with as many people as possible. Initially, I used to be very reserved, but very soon I realized if I am like that, I will be like that only. So you have to make a choice. Okay. So that is one thing.
Yeah.
And then open up
But
yourself. Make mistakes. If you do not make mistakes, you are not going to learn. I will tell you, if anybody says that without any mistake I did it, 99% it must be wrong. Very rarely you will achieve perfection without mistakes. So that is one thing. And no feelings at all if you make a mistake, but that mistake cannot be repeated. Maybe twice, but you have to learn from each mistake. And then attitudes are different. Each individual is different. Don't have expectations from others. Okay, you should behave like this, you should work like this. Who are you to dictate? They have to decide for them. This is the problem where we have conflicts. Okay, it is your expectations. Why do you expect that somebody has to behave like the way you want it? So that is another thing. Coming to the scientific aspects, I think each one know that where they want to go and how to work. But most importantly, your attitude and how you work with the people. In my lab, I make it a practice that I tell everyone this is the actually I say that this is the first house, because you spend good amount of time here, 9 to 6 on every day. By the time you go home, couple of hours you eat and talk to them and you sleep. So that means actually the time that you productive time that you spend is more here. So it is your first house rather than your second house. So then try to make it as a family culture that you are living with your family members. Then things will fall in place. You can fight, but tomorrow you can be all right again. Okay, come back.
Thank you, sir. Thank you so much for the wonderful stories. You have been my inspiration. You have been a guiding force, for many as well. It's been absolutely incredible. The most important thing that we could really learn from you is how to be simple, how to be a kind of a down-to-earth person, and how to learn, even from a very smallest person. You have really acknowledged so many people, and they have really touched your hearts, and many of them also, would be, wonderful mentees for you as well. And I could see, from today's conversation that there are many people who have really asked. As soon as we posted it, there are a few people, who are watching in YouTube as well, because the link is not really sent to all of them.
Okay.
I was just checking. There were about 33 people who were watching in YouTube, which we posted in Bioclues as well, just to
But
create this
one thing,
man.
Prashant, if I may take a second,
Yeah.
my science apart, it's not any breakthrough or whatever it is on par with the world science where we stand is a different thing. But having reached to this stage, one thing that in my mind is to help the people as much as you can, particularly the younger generation. If they want to make a career, what is that can I provide my lab? Can I allow them to do some experiments? And if they want to move from my lab to some other best lab, how much I can help them? Or somebody who wants to make an entry into the research, how do we help them? So that is one thing I am very clear in my mind. Then of course in science wise I have my own limitations, my abilities, how much ever I can do in a best in a given ambience and in a place. We try to do the way in a best possible way, but that is up to others to judge where do we stand. But do your best in a given situation and help others. Because today I am here because of the so many people in addition to my family. I told the wonderful people who gave birth to me, and then my family, my brother's family, my sister's family, and my own family, and then your friends and relatives. I had the enormous support, mental and emotional support from my colleague Reddy, and then many other people. Now I have a colleague in the institute, Raghu. I want to tell you one thing, Satish. And almost every week we talk. Out of 30 minutes we talk, 20 minutes Satish keeps talking about science. That's what I call him is my mentor. So you learn it. He tells me so many things. He says, sir, tomorrow you set up, isolate fibronectin from plasma. I am not yet I'm yet to do. For the last 2 months, he's asking me to isolate that. So this is how it works. Okay. So thank you
Thank you,
again
sir.
extending.
Thank you so much, sir, for your time. So, friends, this is it for today. Sir has given wonderful this thing. It's a great honor. Sir can be reached by his email. If you simply type Bhanu Reddy or BhanuPrakash Reddy, the very first hit is Bhanu sir's page. So please get in touch with him. He's such a lovely person, and please feel free to visit his lab whenever you're in Hyderabad. He's such a guiding force. Yeah, thank you so much, sir. And I'm sorry, if some of the questions were not posed. I felt that there were some questions which are not relevant. So please excuse me for this. Thank you so much. Yeah. Thank you, sir.
Prash, thank you so much. It was a pleasure and nice time. It's a casual talk, like you sit on a coffee shop and then do your casual thing. But we made these 20, 30 people to sit with us. So I don't know whether they enjoyed it or not. Thank you once again.
No, no, thank you, sir. Thank you so much. Namaste, sir. Thank you.